everybody. All right, we have gone through the entire infectious cycle. Previous 11 lectures. You now know what happens in cells infected with different viruses. Now we're going to switch gears. For the rest of this course, we're going to talk about infection of organisms, mostly humans, with some animals thrown in as well. And this is very different, but this is a little more immediate if you're interested in infectious diseases. So here is what we've done the past 11 lectures. We've studied virus replication in cell culture. That's the only way you can come up with the information that I've told you about structure, transcription, translation, DNA replication, assembly, and so forth, by studying virus replication in a plate of cells where every cell can be infected. You can make purified virus stocks. But cells are an artificial system, of course. They don't tell you a lot about what happens in an animal or a human, where there are many, many more things that have to occur. In a dish of cells, we're giving the virus free lunch. They're the cells. They don't have to get to them. In an organism, the viruses have many more things to accomplish. They have to find an organism, they have to get in, they have to replicate and spread, they have to find the right tissues, and most importantly, they have to get to a new host. In my view, that's the most important driver of virus evolution, transmission to a new host. And that depends on a lot of parameters, which hopefully we can talk about in the next, how many lectures? 12 lectures. And it's not just flying through the air with the greatest of ease to reach another host. It's multiplying to high enough titers, for example, in the first host to make sure you can be transmitted, by, whether it be by a mosquito or fecal contamination or an aerosol. Lots of parameters. So it's a lot more complicated in the host. So start to think about that. Get away from the mentality of tissue culture and think about a multicellular host. But all the principles we've learned are still applying. The origin of replication is still where DNA synthesis initiates. DNA viruses have to make at least one protein in order to commandeer DNA replication. All that still holds. We learned in cell culture. But now it's another level of complexity, and we're going to talk about the issues that are involved, which include immune systems. Cells in culture don't have adaptive immune systems. They have innate immune systems, but not adaptive, so that makes a big difference. And there are no blood vessels or nerves. There's nowhere to reach. You just get right into the cell and you multiply. So those are the things we're going to be talking about when we talk about viruses going into their hosts. And today I want to give you a general introduction to some of the principles that govern all interactions of viruses with multicellular hosts. As I said in lecture 1, we live and prosper in a cloud of viruses. As you take the next exam on Monday, you're going to be taking it in a cloud of viruses. And you're full of them, of course, as you know. However, as you also know, and I think I told you, most infections have no consequence. And if we do get infected, that is, if the viruses are replicating in us, often they are inapparent. Inapparent. That word is very important to remember, because it means an infection without any outward symptoms or signs. You know the difference between a sign and a symptom, by the way? No? Well, one of them is what you can tell the doctor. I feel achy or I feel sick. That's a symptom. And a sign is what the doctor measures. And mice do not have symptoms when they're infected. They can't talk. They only have signs of infection. So a symptom is something that can be verbally transmitted to a caregiver. Inapparent means you don't have any signs or symptoms, but the virus is still replicating, and we know that. I'll tell you why we know that in a moment. Here's an example of a virus. Now, every virus pretty much has inapparent infections. Don't let anyone ever tell you that 100% of any virus infection is apparent. There's always going to be some. Some of them are close to 100%, but never 100%. West Nile virus. This is a flavivirus. We've talked about flaviviruses. Immediately should pop into your head. Genome polarity, structure of the virus particle, RNA polymerase, all that stuff should come pouring out as soon as I say flavivirus. It doesn't, huh? I'm sorry about that. it does for me, but then again, I've been thinking about it for 40 years. So this is a mosquito-transmitted infection. Very interesting. It was not in the US before 1999. Came here in New York City. It first arrived in New York City, probably on an airplane from Israel. We don't know. It may have been a mosquito or an infected person. Came to New York City, started an outbreak that was picked up because birds in the Bronx Zoo were dying. Very interesting story. Anyway, that's not what we're going to talk about today. By October of 24, about 1 million people were infected because, and we know this, we didn't have 1 million people come to doctors' offices, but we did serology on a subset of them. We took 1,000 people and we did serology, and we understood that a fraction of them were seropositive. They have antibodies against the virus. We can extrapolate that to the bigger population. But febrile illness developed in 20% of infected people. So 20% of the seropositive people had fever. So those were the inapparent illnesses, the 80% or so that were infected. They're seropositive, so we know they're infected. They have antibodies to the virus. If you do not have antibodies to the virus, you're not infected. And 20% of the infected people develop a febrile illness, which means you get a fever. You know, many viruses, it turns out, cause illnesses which, for the first period of infection, all seem about the same. You get fever, malaise, body aches, muscle aches, variety of other things. We call those flu-like symptoms, but they're not just caused by flu. So that's what we mean by febrile illness. Neuroinvasive illness develops in about 1% of infected people, and that's serious. So many people were infected with no obvious disease. And this is a problem for public health officials, because if you can't tell people are infected, it's very difficult to stop the spread of the disease. That's why we like to vaccinate for viruses that cause extensive infections. Just vaccinate as many people as possible, and we can stop the spread. Of course, to develop a vaccine, you have to have a medical need. You have to have enough people infected. If there are 5 infections a year caused by a virus, this is not possible to make a vaccine against it. Nobody will pay for it. But if you have thousands or millions of people infected, you make a vaccine and then you use it. Now, unfortunately, we're in an era now where there are loons walking around the globe who think vaccines either don't work or they make you worse sick than the virus itself. And it's very hard to combat that, but it leads to people dying from vaccine-preventable illnesses. We'll get back to that later. So that's an example of an inapparent infection. There are plenty more of them as well. Here's how West Nile spread throughout the U.S. 1999 came to New York City, and then slowly New Jersey, other states. You can see 2003, 4, 5, 6, 7, 8. Eventually, it's all throughout the U.S. now. It's in every state. And this is because the mosquitoes brought the virus westward. So the rest of this course, we're going to be talking about a topic called viral pathogenesis. So pathogenesis is a general word which means the process of producing a disease. It's not specific for viruses. You can talk about the pathogenesis of a bacterial infection or of a neurological disease or pathogenesis of cancer. It's just development of disease. But we talk, of course, about viral pathogenesis, and I want you to remember there are always 2 components to the pathogenesis of a disease. There's what the virus does and what you've been learning about up to this point. You've learned that the virus infects cells, it multiplies, and in many cases it kills the cells. So obviously if a virus is replicating in your liver and kills liver cells, you're going to have symptoms, and that's part of the disease. But not every virus kills cells, and in fact in a host we have the added variable of the host response. And for many viruses, it turns out that most of your signs and symptoms are in fact not caused by the virus, but by your immune response, responding to virus infection. The fever, the flu-like symptoms, aches, chills, that's not virus. Especially for flu, take this as an example. Flu is replicating in your lungs, and you have muscle aches in your leg. Now, what's that about? Those are cytokines being produced in your lungs and getting in your circulation and going elsewhere. That's why your fever rises. So the host response is a big deal. In fact, in some of the viruses where they cause no cytopathic effects whatsoever, as we'll see, all the symptoms and all the damage is done by your immune response. So maybe we should just get rid of our immune systems, right? Well, no, that wouldn't work, because then viruses would kill everyone, I think. But remember, virus and host, whenever you look at a disease, no matter what it is, the signs and symptoms are a combination of the two. So we study a lot of interesting questions in viral pathogenesis. I've spent most of my career on trying to understand not only how viruses grow in cells, as we've talked about, but I'm interested in viral pathogenesis, and we've developed models to work on this, as we'll tell you. How does a virus get in the host? How does the host initially respond? Where does the virus first replicate? What cells? How does the virus spread? Eventually, what organs and tissues get infected? Is the infection cleared, or is the virus with you forever? Some viruses stay with you forever, and we're going to talk about that, how that works. And finally, how is it transmitted to other hosts? This is really important, as I've said, and I'm going to keep emphasizing it. If a virus stays in a host and doesn't transmit, that's the end. That's the end of the line for that virus. And so I think the biggest evolutionary pressure on a virus which we can recognize is the pressure to transmit to another host. These are all sorts of questions and more that we study and try and understand why. We'd like to know how viruses cause disease, and we believe it gives us insight into preventing disease either with vaccination or antivirals or other therapeutics. All right, in order for a virus to successfully infect the host, 3 general requirements. You have to have enough virus. Enough is a tough question to answer. It varies depending on the host, the species, whether what the virus is in particular. So in some cases, 1 infectious virus in a mouse is enough to give disease to the mouse, but in humans you may need 100,000 or 1 million virus particles, so it really depends. But you have to have enough. So for a virus that needs 1 million PFU to start an infection, if you acquire 10, you're not going to get infected. So you have to have enough virus. Cells have to be accessible to infection. We'll see some examples where virus never reaches cells that can be infected, so there's no infection. Cells have to be susceptible. Remember what that means? They have receptors for the virus, and they have to be permissive. They have to be able to replicate the virus beyond uptake. And in addition, the third requirement is that the local antiviral defenses, they either have to not be there, which happens sometimes, or they're overcome by virus replication. So we're constantly assaulted with viruses, and our immune systems do pretty well at handling most of them. But in some cases, they're overcome and illness results. And we'll talk about that in the remainder of this course. So let's divide pathogenesis into a few stages, just conceptually, so we can discuss it. The first is getting into a host, gaining access. And if you think about it, there are not many ways that viruses can get in us. And here they are. We have a limited spectrum of entry sites. They're on this picture here. We have skin, which is one site, which we'll talk about. And then there are a variety of mucosal surfaces. For example, your eyes, your mouth and nose, respiratory, alimentary, and urogenital tracts. And that's really it. So the mucosal surfaces in the skin are the ways that viruses can get into us. So let's talk about the skin, the biggest organ in your body. It's an organ. And here's the way it's made up on the left here. The outer layer is called the epidermis. And the very outer layer of the epidermis, the stratum corneum, is a layer of dead cells. You're covered with dead cells. You're walking death, all of you. And those dead cells are always falling off. And a good part of dust in any room is your dead skin cells. It's not all dead skin. There's other stuff too. But if you took some dust from a room and ran it on a gel, you would see collagen, a major protein in skin cells. So it's always falling off, and that's why it's a good defense. The outer skin is dead. Viruses can't replicate in dead cells, right? So that's a great defense. So it's relatively thick. And so a virus just falling on your skin will not be able to infect those cells. Unfortunately, most of us have many abrasions on our skin and cuts and scratches and nail biting and all that. And so viruses can gain access. Mosquitoes are very good at delivering viruses. They go right through that dead layer. So this mosquito here, remember, only female mosquitoes take blood meals because they need that to make eggs. They put their proboscis right through the epidermis into the subdermal tissues where the blood vessels are. And there's a great movie, I'll link to it, of a mosquito looking for a blood vessel from below, looking below the skin, and it keeps probing and probing until it finds one, and it hits it, and then all of a sudden the blood vessel collapses because it's applying such suction to pull the blood out that the blood volume goes down. So they actually look for a blood vessel, and as they're doing that, they are depositing chemicals, including an anesthetic so you don't get itchy and slap them, and an anticoagulant to keep the blood flowing. And then, of course, if they have viruses in them, out come viruses with those chemicals as well. And then when they pierce a blood vessel, the viruses go right into the blood. But the virus doesn't have to go in the blood. It could also go in the subdermal tissues and replicate as well. All right, so below the stratum corneum, the epidermis is then made up of layers of living cells. So if you get a scratch, the virus can get into them and replicate. And then below that, the dermis is then when you see blood vessels and lymph vessels and immune cells floating around. And if a virus can get down there, then it can get into the blood circulation and it spread elsewhere. So the skin is one way of getting in. Mucosal surfaces are the other site. And of course, the skin also, I forgot to mention, besides scratches and abrasions, dog bites, raccoon bites, etc., can give you virus infections, and insect bites as well. So the mucosal surfaces are ripe because they are permeable. They are moist, permeable surfaces lined with living cells, and their purpose is to take in materials and get rid of them. You know, your respiratory tract has to take in oxygen and give off CO2. So they're by design accessible, and unfortunately that's very easy for a virus to get into them. So they'd include the eyes, respiratory, and the alimentary and urogenital tracts. So here's a schematic of the respiratory tract. It is, of course, it starts up with your mouth and nose, the nasopharynx, and then the different parts of the lungs, the trachea, the bronchi, and the bronchioles. And this is all lined with ciliated epithelial cells. And these have a brush border, which is then in contact with a mucus layer. Mucus is produced by goblet cells. The epithelium at the bottom is lined by what's called the basement membrane, which is a very impermeable membrane that prevents things from coming through readily. And viruses can infect these cells and get through. We'll see how that happens in a moment. The mucus is a very good protection against that. It catches a lot of particles, and you have a mucociliary elevator in your respiratory tract. It's lined with cilia, and when particles get in, the cilia and the mucus combine, pushes it back up into your mouth, and then you swallow every few seconds or so, and you're swallowing mucus that has particles in it, and they'll get digested in the respiratory tract. So that's a kind of nonspecific defense. Now, viruses can infect every layer or every level of the respiratory tract, from the upper tract to pharynx, larynx, the trachea, the bronchi, and the bronchioles. These are all different viruses. Some can replicate in both sites, some are specific, but these are the manifestation of infection at these different sites, pharyngitis, laryngitis, down to bronchopneumonia and bronchiolitis. So it's a very good portal. Now, I think we inspire about 6 liters per minute of air. It's a lot. So we're always sampling the air around it. We need the air, of course, to survive, but the air is full of viruses, and so we're always taking them in, bringing them deep into our lungs. So the mucociliary elevator is an important way to get rid of them, and other immune defenses as well. There are macrophages down here in the alveoli as well. The alimentary tract is another one, from your mouth and esophagus through the stomach in the large and small intestine. It's lined with similar epithelial cells that are exposed to the environment. It has to be able to take in nutrients and excrete waste as well. So it's a good portal for entry. As in addition, the contents are always in motion. From the moment you swallow food or drink something, it's in motion in your stomach and your intestines. So it gives good opportunity for viruses to contact cells, and it has to contact cells, of course, on the luminal surfaces in order to infect them. So it's a good place, and we're always eating and drinking, of course, and so it's a good opportunity to introduce viruses. So here's a close-up of the small intestine. Here's an electron micrograph on the right. The small intestine is lined with ciliated epithelial cells as well. Sorry, just epithelial cells. Let's start with the diagram on the lower left. Cross section of the small intestinal wall. Here is the epithelial cell sheet, which forms the surface. And in the exposure on the top here, the magnification, you can see the epithelial cells, or enterocytes in this case. They're specialized on either side of what's called an M cell. The M cell is important for sampling the gut lumen for foreign antigens, and so immune cells will often, like dendritic cells, will often reach through and sample antigens. And it's a good way for viruses to get into the underlying tissue. These cells are also lined at the bottom by a basement membrane, which protects things from getting through very easily, but cells like macrophages can readily go through those. Here on the right is an electron micrograph of an M cell surrounded by enterocytes. So the gut, of course, is a selectively permeable barrier. It has to let foods, digested foods in and release wastes. And it's obviously, just like the respiratory tract, in direct contact with the outside world. Urogenital tract is another example of a mucosal layer that's exposed to the world. It is also protected by mucus and low pH, but abrasions can let virus into these cells that line the urogenital tract. And we know a number of viruses that infect there, for example, the human papillomaviruses, which can cause warts, anogenital warts, and cancers. Also, HIV and herpes simplex viruses infect the urogenital tract. They can spread elsewhere, as we'll talk about later. And finally, the eye is another portal of entry. You're always blinking, and that's a way to prevent things from getting in your eye. Your eye is bathed in fluids. You blink every few seconds. The fluids wash away any grit or debris or viruses and bacteria that might be caught there. But occasionally viruses do infect the eye. When they do so, they primarily infect the membranes lining the eye, the conjunctiva, and the sclera. Those have the red asterisk. Some viruses, like enteroviruses, do that, and they cause subconjunctival bleeding, which you may have experienced in your life. This is not harmful, just looks terrible, but it could be caused by a virus infection. As we'll see later, some viruses can cause blindness by causing problems in the cornea. And the herpes virus is a major cause of blindness globally, that do so not by infecting the cornea, but by infecting the sclera and the conjunctiva, and causing an immune reaction in the cornea that causes blindness. So the eye is another portal. If you have a hot tub or a pool that's not properly sanitized, viruses can be existing in it, and many eye infections are caused by people who go into those bodies of water and they get viruses in their eyes, end up with conjunctivitis. Now, the fetus is really another organ that we should consider when we talk about where viruses go, and this has been in the news a lot lately, which is good, but fetal infection is nothing new. We've known about this for many years. So the fetus, the developing fetus, can be a target of virus infection. No matter how the mother acquires the infection by any of the routes we've talked about, it could reach the fetus. That's transplacental infection, when there's a baby in developing in utero and the mother gets infected and the virus crosses the placenta and infects the fetus. We call that transplacental infection. That's in contrast to perinatal, where the baby acquires infection when it's born from all the blood that's present during childbirth. HIV very commonly acquired during birth, not transplacentally. We have identified the so-called torch pathogens, Toxoplasma, rubella, cytomegalovirus, HIV, and others that cause birth defects. And the O stands for others. They were very smart to put an O in the name because they knew that more and more agents would be identified. And there are other viruses that can cause transplacental infection. And of course, Zika is one of them, and we'll talk about that later. All right, first question. The outer layer of which of the following is dead but can still serve as a portal of virus entry? Respiratory tract, alimentary tract, eye, skin, urogenital tract. The answer, of course, is D. I was hoping you'd get 100% there. Yeah, if your outer layer of your respiratory tract is dead, then sadly so would you be. All right, so that's how viruses get in general. We'll talk some more specifics later on. Let's talk about spread from that initial site of infection. So there's a limited number of sites that viruses can get in. and many of them stay there. So many viruses enter the respiratory tract and stay there and cause disease. They're respiratory pathogens, and we call those localized infections. The virus infects an epithelial cell, spreads laterally, you get a respiratory infection, but some spread. Some are disseminated, we say, when a virus spreads from the initial site of infection, whether that be the skin, eye, respiratory, alimentary, or urogenital tracts. And when many organs are infected, we call this a systemic infection. Now, for viruses to spread from the initial site of infection, physical and immune barriers have to be overcome. So what do I mean by that? So let's take the respiratory tract here on the right. Nice layer of ciliated epithelial cells, a mucus layer, and most importantly, a basement membrane, a membrane underneath, which is it's relatively impermeable to virus particles. So how do we get beyond that? If viruses are replicating in these cells, how do we get beyond it? The same issue occurs with intestinal cells. There's a nice layer of enterocytes. Well, it turns out that in the intestine anyway, the M cell is a good portal of entry, as I've just suggested. Its job is to allow things to come through, so many viruses bind M cells and get passed through them by transcytosis. Sometimes they're picked up by immune cells that are sticking through. You see some immune cells like macrophages have a way of getting through the basement membrane. They can disrupt it. So the basement membrane can be disrupted by cytokines that are produced in response to virus infection. That's called inflammation. So inflammation loosens up the basement membrane. Why? So that immune cells can get through, and this can be in the gut or the respiratory tract, and try and clear the infection. Of course, the downside is that viruses can then get through the basement membrane. And in this picture of our respiratory tract, the same thing can happen. If you have virus infecting these ciliated cells, which happens all the time, the right cytokine response, that is the production of immune molecules by the innate immune system, can cause the basement membrane to loosen up, to lose its integrity, with the idea of letting immune cells through, but then viruses can get in as well. So it's a very nice example of how the immune system is a double-edged sword. And we will talk about later how that's balanced in infection. So viruses can breach the basement membrane in some cases by inflammation. The other key variable in spread of viruses from the initial site of infection elsewhere is where the virus is released from cells. Now, these epithelial cells, whether they be in the respiratory or the gut tract or wherever, they're polarized. They have an apical and a basolateral domain, which are very different. They have different proteins on them, and they're structurally very different. And some viruses only are released at the apical domain of cells. So, for example, here's influenza virus. It's only released at the apical domain of polarized epithelial cells. And so that makes sense. You cannot spread. So a virus that is not, you can't spread unless you're released from the basolateral domain after replication in the cell. So a virus like influenza released at the apical domain, not likely to be able to spread. And in most cases, we know flu viruses do not go systemic. Here's measles virus, an example of another virus which is only released apically as well. Now, turns out that measles does get into your blood by different mechanisms that don't involve release, so we're going to talk about that later. And here's VSV. VSV is released at the basolateral domain, so that's a virus that has the potential to cause systemic infections, to be able to spread beyond the initial epithelium. Now, here's a good example of how directional release of virus can control pathogenesis. There's a paramyxovirus called Sendai virus. It's a model organism. It infects mice, doesn't infect people. The wild-type virus is a respiratory pathogen. You can inoculate mice by aerosols. The virus is released apically and only remains in the respiratory tract, never spreads. You can make a mutant of the virus. You can alter the viral glycoprotein in one or two amino acids. So now it's released both apically and basolaterally. What do you think is the pathology of that virus in a mouse now? It can spread. It causes disseminated infection simply by virtue of it being able to be released from the bottom of the epithelium. Now, it's not always going to be all that's needed, but it illustrates nicely how directional release is really important for spread from the initial site. All right, so we have a body surface epithelium. We have the virus getting across it by being released basolaterally and also by having inflammation that disrupts the basement membrane. And now the virus is in what we call subepithelial tissues. That simply means below the epithelium. And they are full of blood vessels and lymph vessels and immune cells. And there it lies the ticket to other sites for the virus. Many viruses then enter the lymph system. So, besides having blood vessels and capillaries throughout your tissues, you also have a lymphatic system. And there are capillaries as well as lymph vessels throughout your body. And the capillaries tend to be more permeable than blood vessels, so viruses typically get into them. Then the lymph system, of course, is made up of lymph nodes, central parts of the immune system. And the viruses can go from the lymph nodes and eventually reach the blood because the lymph and the circulatory systems are connected. Now, viruses can also get into capillaries. A mosquito could deliver a virus to a capillary, or a virus could even get in other ways. But the lymph system is a major way to get access to the blood. And now the virus can go anywhere it wants to, right? Because the blood circulates to every cell and organ in the body. And blood in the virus in the blood is called viremia. And this is a graph that shows you what is a viremia. So we're looking at infection of an animal with time, days after infection, and we're looking at virus titer on the y-axis. And we distinguish three kinds of viremia. Let's say we took a mouse and we put in the tail vein a little bit of virus, 100,000 PFU of virus. If we then took blood immediately from that animal and titrated the virus, you would see some virus in the blood. That's simply the stuff we inoculated. It hasn't replicated yet. We call that passive viremia. That's just what we put in the animal. Eventually, and how long it takes depends on the virus in the animal, that virus will replicate somewhere. And the initial burst of virus replication is typically where the virus has landed, the primary sites of infection of the virus. And that's the primary viremia. So it's sort of like virus getting into a respiratory tract, initially replicating at the tract, at the mucosal epithelium, then spreading to the blood. That would be the primary viremia. Once the virus is in the blood, it can reach any other tissue or organ. When it finds one that it can replicate in, it replicates there, and that produces a second burst of viremia, which we call the secondary viremia. All right, so we have passive, primary, and secondary. The primary is replication at the initial site of entry. The secondary is from distal sites from the initial site of entry. And the distal sites are seeded by virus in the blood. Now, viremia, of course, is really important because it spreads viruses beyond the site of entry, and that's of concern. Viremia is how we diagnose some viral infections, so it has diagnostic value. But of course, it's a problem for the blood supply because we haven't yet learned how to make synthetic blood, so we need donor blood. And when people are sick, they have viruses in their blood, and many, many, many virus infections have been spread with contaminated blood before we learned that it was containing a certain virus. And the numbers are huge over the years. HIV is a big one, which we didn't know of soon enough to stop transmission via the blood, but hepatitis viruses of all sorts. And until we discover a virus and realize it's in the blood, develop a test for it, we can't check the blood supply for it. So right now, blood is checked for many, many different viruses, but guess what? There's still viruses in every pint of blood that is donated. If you threw away blood because it had any virus in it, you would have no blood supply. And that's because you are all infected with a lot of little viruses that don't do you any harm, and so we've decided, well, if we want to have blood, we need to be able to keep those. So there's no evidence that they do any harm. But we do test for HIV, all the hepatitis viruses, and many other viruses as well. So let's look at a classic virus infection of mice to get a sense for how this whole story that we've outlined so far fits together. This is the pathogenesis of mousepox. A mousepox virus is related to smallpox virus, the scourge of humans until it was eradicated in 1979, and people studied it in mice. This is a mouse version of the virus, And this is a naturally occurring virus of mice that causes a rash, very much like smallpox. And these viruses are initially, in the laboratory, you can inoculate them into the foot pad. And that is actually very much like the way the mice get infected in nature. They walk around, right? And the virus is on the ground. It's excreted by mice, and their foot pads are nicked by the rocks and whatever they're walking on and the sticks. And the virus gets into the nicks on their foot pad and infects. So that's a very nice route of experimental infection. So you infect the virus, you put the virus in the foot pad. That's basically a sub-epithelial inoculation. So the virus immediately has access to the lymph system, which you see, that's a lymph vessel, and there's a lymph node. And the virus can actually replicate in cells of the lymph node and makes its way into the blood. So we have our primary viremia. So a little local replication at the site of inoculation and then spread to the blood. The virus is then carried to the spleen and the liver, where it multiplies further. It destroys those organs. You get what we call necrosis or tissue destruction. And then replication at those sites releases more virus into the blood. That's your secondary viremia. And then the secondary viremia brings the virus to the skin. And eventually that causes the rash that appears on the mice. So that's a result of secondary viremia. When the rash arises, the foot pad also swells. That's the original site of inoculation, and this is an immune response to the presence of virus. So this is a classic experiment or series of experiments done by Frank Fenner, an Australian virologist, who for the first time showed how viral infections proceed, from local multiplication to primary viremia, seeding of secondary organs, secondary viremia, and in this case, the skin. And we'll see how this applies to human infections as well. So here's a big slide, lots of information on it, which essentially tells you the same thing, but for different viruses with different kinds of targets. And we start at the top here. We have infection. It can be skin, mucous membranes of the respiratory or gastrointestinal tract, any of the places we originally discussed. That's your initial infection. And there's virus multiplication there at the site of infection. Now, many viruses are shed from that primary site of inoculation, like herpes viruses and papillomaviruses, and that's how they can infect others right away, shortly after infection. Then the virus moves through the lymph nodes, via the lymph nodes to the blood. You have your primary viremia, and then that viremia can bring it to different targets, and the targets depend on the virus. Not every virus infects every organ, like muscles and liver, spleen, etc. Viruses can even infect the cells of the blood vessels. Then we have our secondary viremia, and that brings it to different sites. And then typically the characteristic disease of the virus involves multiplication at these secondary sites. So for example, poliovirus would be reaching the brain and spinal cord and causing paralysis after a secondary viremia. And here at the bottom are different viruses that target different organs. Rotaviruses would target the GI tract and cause gastroenteritis. So it's a nice summary of the different ways that viruses can spread. You don't need to know this for each virus. I want you to understand the concept of entry, primary replication in viremia, primary and secondary viremia, and spread to other organs. Now, some viruses cause a rash. Not all of them do, and I want you to be familiar with the words that are used to describe rashes. And here are some viruses that cause skin rash, and of course, Zika virus is the latest addition to this. We've known that Zika virus causes rash-like illness since 1947, but most people didn't know about it. Now nearly everyone does. But other viruses, Coxsackie viruses, measles, of course, the parvoviruses, those single-stranded DNA viruses that we talked about, cause rashes in kids, rubella virus, chickenpox and shingles virus, and Zika. And here we have two different kinds of rashes. So obviously this is a consequence of the blood bringing virus to the skin, typically via a secondary viremia. And only certain viruses will cause a rash. It's typically an immune reaction in the skin that's causing this. I have two different kinds of rashes listed here. One is a vesicular rash, and that's chickenpox or shingles, and that's on the right here. And you can see the individual rash lesions are vesicle-like. They're full of fluid, and they are raised from the skin. quite clearly vesicular. Then we have maculopapular rashes, which is the one on the left. This is a mixture of slightly elevated rashes, like the ones here, and flat red ones, which you can see some of the other ones. Maculopapular. There's a few other names as well, but the point is that some viruses can do this, and they have distinct presentations. Next question. In general, secondary viremia is a consequence of which of the following events? Viral replication in the bloodstream, viral replication at the original site of entry, viral replication in organs distal to the site of entry, viral replication in lymph nodes, all of the above. All right, the answer here is viral replication in organs distal to the site of entry. That's secondary viremia. Now, I could understand if you're confused by A or, well, viral replication at the original site of entry is not correct. That's primary viremia. So it can't be all of the above. And the other one, replication in the bloodstream, which I didn't mention at all. So technically that could be true. So you can imagine that there's a primary viremia that seeds immune cells in the bloodstream. But the key point I want you to understand is that they're typically distal from the original site of entry, the secondary viremia caused by infection at other sites. We've talked a lot about virus spreading through the lymph and the blood. There are other ways that viruses can spread, and one of them is neural spread. And that's illustrated on this slide. So here on the left is a diagram of both sensory and motor nerve endings. So of course, these are wired back into the spinal cord. So viruses can enter motor muscle nerve endings or sensory nerve endings and travel up the muscle fibers all the way into the spinal cord. This is clearly what happens with poliovirus. It establishes a viremia. The viremia, of course, brings it to the muscle, where the virus can then attach to receptors at the end of nerves and muscles and travel up to the spinal cord in the brain. And herpes viruses are other viruses that do this very well. They tend to stay in peripheral nervous system, like these ganglia here. Now, this movement, of course, isn't by diffusion. It happens by motor proteins on a cytoskeleton, and that's illustrated in the upper right of this figure here. Here is a cell body of a neuron and a neuron terminal. You know, it could be a sensory or motor terminal here, but viruses enter the terminal, they go towards the cell body, and they're moving on synaptic vesicles. And here's a herpes virus particle inside a synaptic vesicle, which was formed at the nerve terminus, and it's being brought up the microtubules, in this case by kinesins, towards the cell body. So that's spread, that's retrograde spread towards the cell body. And then, of course, there's a dynein motor that brings virus the other way. We talked about this in cells as a way of getting adenovirus towards the nucleus, and then the dynein motor starts to pull it away once it's docked on the nucleus, and it breaks the particle open. So viruses travel in nerves by motor pathways. It is not diffusion. Poliovirus does this as well, herpes viruses and some others. This ability of viruses to do that has actually been used to map circuitry in the brain. So you can take herpes viruses, which are very good at traveling in nerves. You can introduce a green fluorescent protein gene in them and then place them, inoculate them with a needle at very specific places in the central nervous system, stereotactic inoculation, and then you can trace the nerve pathway. So you can put them in a specific part of the brain and answer the question, what's wired to that part of the brain? That's what's shown here. Here, the virus was inoculated at one place, and you can see all the neural connections that are made through synapses. So we have nerve cells and fibers that are synapsed together, and the virus can travel from one to the other. It crosses the synapse, so it infects the cell body, it's released, crosses the synapse, and enters another nerve. So you can use this to map the connections in the brain. It's pretty cool. Now, when a virus infects the central nervous system, as I'll emphasize often in this course, I think for the most part, virus infections of the CNS are accidents, and I'll give you some support of that statement later. But we have certain terminology that we're going to use, and they're on this slide. I want you to understand them. So a neurotropic virus is a virus that can infect neural cells. Doesn't matter how it gets to the brain or spinal cord, as long as it can infect neural cells. In fact, if you make neural cells in culture, and the virus can infect them. It's neurotropic. So it's independent of the way the virus gets to the central nervous or peripheral nervous system. A neuroinvasive virus can enter the CNS when you inoculate it peripherally. So if you put a virus in the footpad of a mouse and it ends up in the brain, that is a neuroinvasive virus. Now, we're not saying that it's neurovirulent. We're not making any implications about the ability of the virus to replicate in the brain. It just gets there. Neurovirulent viruses cause disease. Again, no implication of whether it's neuroinvasive or assuming it's neurotropic, because if it can cause disease, it's probably replicating in nerve cells. So let me give you some examples. Herpes simplex virus has low neuroinvasiveness. So in most herpes simplex infections, the virus remains in peripheral ganglia. And we'll see when we talk about this in detail. Periodically, the virus comes out of the ganglia and goes to your mucosal surfaces, makes a fever sore, for example. But in very rare occasion, it goes the wrong way and goes into the central nervous system. So it doesn't do that very often, so we say it has low neuroinvasiveness. But when it gets there, it's highly neurovirulent. These are very, very serious infections when herpes gets into the CNS. Mumps virus. Mumps is a virus that used to infect every kid in school age. I had mumps as a kid, and then we developed vaccines, so nobody gets mumps anymore unless you don't get vaccinated. But it turns out that in 50% of kids with mumps, they had virus in their brains, but they were fine. So we say mumps has high neuroinvasiveness. It readily gets into the brain from the respiratory tract, but it's low neurovirulence. It does not cause disease. And finally, rabies, high neuroinvasiveness and high neurovirulence. And this is a bad virus. Fortunately, we have a vaccine, but if you get inoculated with rabies virus, unless you're vaccinated, there's almost 100% chance it will get into your CNS. And once it does that, it's highly neurovirulent. So it's a really bad virus. Very few people have survived, as far as we know, rabies infection without being vaccinated. But the good news about rabies is it takes so long to get to the CNS that you have time to vaccinate after an animal bite. So we have three ways of viruses spreading: lymphatics, blood, and nerve spread. Now, the other issue that I want to talk a little bit about is when a virus is in the blood, how does it get into the tissue itself in order to infect the tissue? We call this tissue invasion. And here are three different kinds of tissue architectures to think about. These are basically the way the blood vessels are juxtaposed to the various tissues. And they're all different kinds of tissues. Here on the left is a capillary and central nervous system, connective tissue, various muscles. Here, blood vessels, of course, are made of cells. They're called endothelial cells. They have a nucleus and a cell body. And in certain tissues, the endothelium junction is very tight, and there's a basement membrane around it. You can see that in yellow here. And so it's very difficult for viruses to get across these. They can do it, but it's not easy to do so. So the underlying tissues are well protected. The CNS in particular is very well protected. I'm going to show you in a moment a more detailed look at this. Then in the middle we have another kind of architecture where there are pores in the endothelial cells. So this blood vessel is made up of two endothelial cells, and there are pores in between them, and viruses can get through those, but there's still a basement membrane. So in order for traversal of that to occur, you have to have inflammation. You can have local infection of the endothelial cell, for example. That can cause release of cytokines, inflammation, loosening of the basement membrane, and the virus can get across. And finally, in some tissues like the liver, there really are very few barriers between viruses getting out of the lumen of the blood vessel. So here we have three endothelial cells making up the lumen of the blood vessel, but they're not even joined together. And right outside of them, there's no basement membrane. If this were the liver, these would be hepatocytes. Now, this is designed this way for a purpose, because the liver has to be taking up material and detoxifying it and so forth. So it's a very easy target for viruses, and hence we have a lot of viruses that can replicate in the liver. Now, that's not, most viruses, if you inoculate them into the blood, say one of the first places they accumulate is in the liver because it's a nice filtering organ, but many of them do not replicate there because they don't have receptors or they're not permissive or whatever. But liver is very easy for viruses to traverse. Now the junction between a capillary and the brain is called the blood-brain junction. You may hear it called the blood-brain barrier. Okay, it is a barrier of sorts because you don't want anything getting into the brain, in particular viruses and bacteria. But it's very well designed to prevent things from getting in, but viruses do get into it. And these are some of the ways that viruses can get through. Again, we have very tightly junctioned endothelial cells and a basement membrane. But viruses, there's the little red dots, they can infect the endothelium. And if they do so, they'll cause inflammation. That'll loosen up the basement membrane. They can be brought across the endothelial cells by transcytosis. They bind a receptor. They're taken into a vesicle. The vesicle fuses at the other side and releases the virus without even replicating. And then sometimes immune cells like lymphocytes or monocytes can be infected, and they can bring the virus into surrounding tissues because they have the ability to traverse the basement membrane. They can elaborate cytokines that loosen up the basement membrane and allow them to get in. Now, the blood-brain junction, I prefer to call it, has been the topic of a lot of study in virology lately because people are trying to understand how viruses get across and how the cells regulate that traversal. Let me show you another slide here from a review article in the Journal of Virology, which just elaborates on this blood-brain junction. So here's the blood vessel again. It's made up of the endothelial cells lining it. And at the bottom is a blow-up of the junction between the endothelial cells, just to show you how complicated it is. It's a very, very tight junction. So it's made up of tight junction proteins, cludins, claudins, JAMs, ESAMs, all sorts of proteins that link the two cells together and make it very difficult for viruses to get in. Now, you may remember that when we talked about, what was it, Coxsackie virus infection. Coxsackie virus receptor is in here. It's the CAR molecule, and a virus cannot access it until the junction is loosened up. So these are very, very tight junctions. There are also other kinds of junctions here called adherence junctions. Then around the blood vessel and around the endothelial cells is the astrocyte. So astrocytes are a certain type of brain cell which are increasingly being recognized to play important roles in immune defense. Their processes surround the blood vessel. And you can see it's made up of different cells. So there's a cell body there, and then its processes are wrapping around. Then there's another cell body at the bottom, and then a third one there. And those are also joined by tight junctions. And so there are multiple layers preventing a virus from getting out. But again, if the virus is causing inflammation, if it's infecting the endothelial cell or an immune cell that's in the blood vessel, it can loosen up all of these junctions. And very recently it's been shown that one of the functions of the astrocyte in response to virus infection, so say a virus gets into this part of the brain, it's infecting a cell, cytokines are produced and the junctions are loosening up. The astrocyte will detect that, secrete interferon, and what that does is close the junction back up again. So not to let viruses into the CNS. So there's an interplay. You open up the junction to let immune cells in, right? But then keep it open only for a while to make sure that the viruses don't get in. It's really, really amazing stuff going on. All right, so this is a summary of the different ways that viruses can get into the CNS. Viruses can get in by nerve spread, as we've talked about, from peripheral nerves. They can get in through blood vessels in the cerebrum, even though they're tightly junctioned. I've told you ways that viruses can get across that. They can be in the cerebrum or in the meninges. Depending on where the virus gets in, it can cause meningitis or encephalitis. And some viruses can even get in through the cerebrospinal fluid. So there are plenty of ways. Now, amazingly, most viruses don't get into the CNS. It's really well protected. But the ones that do cause a lot of damage, and we're going to be talking about some of those later on. All right, finally, tropism is what tissues are infected. Even though a virus may get into a lot of places, most viruses don't replicate everywhere, and where they do is called the tropism of the virus, the spectrum of tissues. And we have words like enterotropic, neurotropic, or hepatotropic to describe where viruses are replicating. Some viruses can be limited. They only replicate in one or two places. Flu, the respiratory cells lining the respiratory tract. Other viruses Ebola virus can replicate in many, many different organs and cell types. Now, what determines that is very interesting. We'd like to figure it out because if you find a tissue that doesn't support replication, maybe that gives you some therapeutic intervention ideas. And some of the determinants of tropism include susceptibility. That's an easy one, whether they're receptors for the virus or not. Permissivity, can the virus replicate in the cell? For example, some viruses have promoters on their DNA genomes that require specific transcription factors, and they may not be present in every cell. That simply will determine where the virus can replicate. Accessibility, some tissues are simply not accessible to virus. Even though it's in the blood, it can't get to it. There are some very privileged sites, and one of them is the interior of the eye, where it's really hard for viruses to get to. And immune defenses. So immune defenses also determine tropism. The tropism of poliovirus, which is limited to the gut and the central nervous system, is determined by interferon defenses. So here's an example of tropism of influenza virus determined by cleavage of the hemagglutinin. So remember, the hemagglutinin is the glycoprotein on the virus particle. It attaches to the cell receptor. And remember, it needs to be cleaved in order for the virus to be infectious. It has to be cleaved right N-terminus to the fusion peptide, so that when the virus infects a cell and the pH drops, the fusion peptide will pop up and enable fusion. I don't know if you remember that, but that's why fusion is needed. The enzyme that carries out this fusion is an enzyme that's only present in the respiratory tract. It's produced by cells called club cells, and here these little Pac-Man things are the enzyme. And as influenza virus is being produced from the respiratory epithelium, it's cleaved extracellularly, the HA is cleaved, and now these viruses are infectious, and they can infect other cells in the epithelium. Now let's say some of these viruses got into your bloodstream and went to your kidney. They would attach to a kidney cell, they would infect and uncoat, because the low pH would uncoat them. You go through the whole replication cycle. The new virus is produced would not have cleaved HA because there's no tryptase in the kidney. So that would be the end of the infection. So that is what limits flu to the respiratory tract. That's one example. There are many, many others, but the point here is that multiple stages of the infectious cycle can limit the tropism of infection. Most influenza viruses, their tropism is limited by this enzyme, which is only in the respiratory tract. Now, there are some highly pathogenic avian influenza virus strains. They're called H5N1. They infect mostly birds. They cause big problems in bird farms, but occasionally they get into people. And then they don't just infect the respiratory tract. They go everywhere. They infect kidneys and liver, et cetera, and cause really, really serious disease. The reason is that they have a sequence difference near the fusion peptide. It's called a multi-basic insertion. And that site is now cleavable by proteases that are present in every tissue in our body. Obviously, this is important for me to have told you because it illustrates how you can change the tropism by just putting in a cleavage site here that can be cleaved by an enzyme that's in all tissues. So that's why we're worried about these H5N1 viruses, because they can apparently replicate in lots of tissues, and they're lethal. They're about 50% lethality associated with infection. Insertion of multiple basic amino acids at the HA cleavage site allows influenza virus to infect many organs. This means that the what of the virus has been changed. Susceptibility, tryptase, clara, permissivity, tropism, all of the above. So the answer is tropism. The insertion of multiple basic amino acids changes the tropism of the virus, and so that is the answer. It's not tryptase class. It doesn't change the enzyme that actually cleaves it. It changed the tropism of the virus. All right, a few more points to end up here. Infections have to be transmitted from one infected organism to another. That doesn't happen in a tissue culture dish, right? Well, you pick up some virus and put it into another dish. That's easy. But there has to be a chain of infection, we call it, to maintain the virus in the population. Otherwise, it's gone. We see two general patterns of transmission, host to host, like human to human. Most human viruses go this way, not all of them, but many do, or animal to animal. And then there is transmission by a vector of some kind. Here it's a rodent and a virus is being carried by a tick, but it could be a human and the virus is carried from human to human by mosquito. It could be a monkey in the forest and the virus is carried by a mosquito. You get the point that there's an intervening vector, human to human or vector, animal, vector, animal. Those are the two general patterns of transmission that we identify. Now, there are also terms associated with transmission that you should be familiar with because we'll use them and you should know what they mean. Horizontal, between members of the same species. So if I'm infecting you by talking and spewing out viruses, that's horizontal transmission. If you're getting infected by a bat, that's a form of horizontal transmission, but we call it zoonotic transmission. And sometimes we can infect animals. Did you know that? We can give our dogs, we can give, actually there's a good example of monkeys in zoos who are infected by human respiratory viruses. That's also horizontal. We call it reverse zoonosis. And then there's some different kinds of horizontal transmission. There's no name for infecting students in a classroom. It's just horizontal. But if a healthcare worker infects you, if you go to a doctor's office and they give you a contaminated needle, that's called iatrogenic infection. Nosocomial, you get infected in the hospital. That happens a lot. In fact, the best thing you can do is not get sick, because once you go in a hospital, it's very bad. Your odds go way down because you get hospital-acquired infections or nosocomial infection. You know, when students take this course at the end, they tell me they don't eat, they don't interact with anyone, they don't go out, so I'm sorry to do that. Vertical transmission, parent and offspring. So at birth, perinatal, that would be one way, but also if your father kisses you and gives you a virus, that's vertical as well. And then germline, when the virus is transmitted as part of the genome. So the retroviruses that we have in us are transmitted that way. Now, most of the time, but not always, you need shedding a virus to transmit it to someone else. And depending on where you're infected, you can be shedding virus in respiratory secretions, mucosa. So often mucus has virus in it, and if you contaminate your hands, you can spread it to others. Urine, semen, feces can have virus. Skin lesions can shed virus. Now blood, you can shed blood if you're cut, of course, and spread infection that way, but the blood supply is an example of how you're not really shedding virus, but they're taking blood from you and can spread virus. In the case of arthropods picking up blood, you're not really shedding it, so they're taking it from you. So insect vectors, germline transmission, those are examples of where you actually don't have shedding, but you still have transmission. Vertical transmission is an example of shedding because the virus is present either at birth or in secretions. So the point here is that most transmissions are from shedding, but not all. And I think that's very obvious. Virus shedding in respiratory secretions is very common. When you sneeze, you give off a big cloud of droplets of different sizes. In fact, you don't even have to sneeze. You can just talk and you give off droplets. And people have studied these. They're different sizes. They give them different names. And the big ones fall right at your feet. And the small ones, the smallest ones can go up to 160 feet. So some of mine can go back there to the back of the room because they're very tiny and they float in the air and they have lots of virus particles in them. So just because you're there doesn't mean you can't be infected by someone else sneezing here. I always think of that when someone sneezes in a lecture, that person in the front could infect everyone in the room. Nasal secretions contaminating hands and tissues also can spread virus infections. They can be everywhere. Viruses can also be spread from skin lesions. Here's a good example of that. We no longer immunize against smallpox because it's been eradicated, but the military is immunized in case some country uses smallpox virus as a biological weapon. So every military recruit is immunized, and they tell you, they put a Band-Aid on your immunization site. The way they do it is they scarify your skin with a needle that has virus in it, then they put a Band-Aid on it, and they say, don't take it off. But unfortunately, some people have sex shortly after getting their smallpox vaccine, and they transmit the infection to their partners. So the Band-Aid comes off, there's virus right on the skin because they scrape it with a needle, so it's replicating right there. And of course, someone touches it, And this virus loves mucosal surfaces, okay? You can figure out where it replicates, but here are two cases in the CDC publication, secondary and tertiary transmission of virus after sexual contact with a smallpox vaccinee, and again here a military vaccinee. So these are all military people, and you have to wonder, if they tell you to keep the Band-Aid on, why can't you listen? Are these good people to be defending our country? So anyway, when you get a vaccine, you get these lesions at the site of vaccination, and they shed virus, and that's how it's transmitted. So I have still my smallpox scar from when I was immunized as a kid, because back in the 50s, we still used to immunize, but no longer. Which statement about transmission is not correct? All virus infections are transmitted by shedding. The route is determined by the site of virus shedding. Transmission is required to maintain a chain of infection. Speaking can produce an aerosol that can transmit infection. Horizontal transmission is among members of one species. Which one is wrong? So the wrong statement, of course, is all infections are transmitted by shedding, right? So mosquitoes picking up virus from the blood, it's not really being shed. Germline transmission is not being shed. Virus in the blood supply, and so forth. Horizontal transmission is among members of one species. I transmit virus to you, that's horizontal. If I get a virus from infection from a bat, what is that called? Zoonotic. All right, a few points to end up. The outbreaks of viral disease are controlled by geography and season, among other things. So geography may restrict the presence of the virus. That may be because there's a reservoir that's restricted, or a vector. So if the reservoir is a bat, only, for example, Ebola is only in certain places because the bats that carry it are only in certain places. So that's geography. And here's a good example, chikungunya virus. And a great example that we've been able to follow in real time of how the vector can affect localization. Chikungunya is a toga virus in the alpha virus genus. We've talked a little bit about before, plus strand RNA virus with an envelope and an icosahedral capsid, nucleocapsid around the genome. It's spread by Aedes aegypti mosquitoes, and it causes rash, fever, and joint pains. We've known about this for many years. It used to be found only in Asia and Africa, these blue areas here. Never in Europe, never in the United States. In 2004, outbreaks began to spread from Kenya to India. One of the first outbreaks was in this island here, Réunion, a French island, where almost all the population got infected. They had never been infected before. Then it spread to Italy, and a lot of countries in Europe have been infected. What happened to change the distribution? Well, first of all, the mosquito changed. The virus was then spread by Aedes albopictus, a different host for the virus. And the original virus didn't replicate very well in Aedes albopictus, but it sustained a 1 amino acid change in the viral glycoprotein, and that now made it replicate really well in this new mosquito, and that's why it spread to other places. So you change the vector, now you expand the geography of infection. Now, Aedes albopictus was introduced into the US by the used tire trade. You know, you throw out tires, people buy them to use them. They put them on big container ships and they move them around the world. They're full of water. You cannot get water out of tires. Think about it. There's no way you can hold a tire and not keep water from getting into it. And don't say cover them, because that costs too much money. So they bring them on container ships open. They're full of water. And it's known that the first Aedes albopictus were brought into the US in Houston many years ago, and they spread throughout the country. Here's now the range of Aedes albopictus. And so now we have some chick in the US. They're all imported, no local transmission yet, but eventually there may be some local cases because the range of Aedes albopictus is changing. Certainly in Europe, they have plenty of Aedes albopictus in the warmer countries, shown by the red here, and that's where they have outbreaks of chikungunya infection. So the vector is very important. We have Aedes albopictus here in New York City. All 5 boroughs have known colonies of Aedes albopictus that are followed. I heard a talk by a New York City entomologist. No Aedes aegypti, though, in New York City. Finally, the other issue that's very interesting is that virus infections are controlled by season. We see distinct seasonality in different virus infections. Rubella, for example, in the US, pre-vaccine, there were regular outbreaks at certain times of the year. These are different months of the year. You can see very seasonal replication. Influenza is, of course, in this temperate climates, a wintertime disease. I told you the other day, this is the prime season now for flu. Even polio is seasonal. You can see, depending on the latitude, it occurred during different times of the year. Here in the US, it occurred in the summer months, but in other areas of the world, it was different. For the most part, we don't really understand this. We don't know why seasonality occurs. There's some suggestion that influenza seasonality is controlled by humidity. So in the winter, it's not very humid. It's very dry and cold, and we think that might preserve viral infectivity. In the summer, it's humid, and under those conditions, at least in the lab, humidity and high temperature tends to inactivate that virus. But that by no means explains all of seasonality of influenza, and for all of these other viruses, we really don't understand what controls why they infect at certain times of the year.